Boa tarde a todos. Vamos iniciar nossa última sessão da tarde. Queria convidar todos a ocuparem seus lugares. Eu sou Cláudia Gomes Padilla, sou médica do Corpo Clínico da Huntington e co-diretora médica em São Paulo. E eu gostaria de chamar a primeira palestrante da nossa sessão, a doutora Tiffany Stankewicz. A doutora Tiffany is a fully trained embryology and the Global Endometrial Product Director for Igenomix USA. Her lecture is titled Optimizing Implantation from the Endometrial Point of View. Welcome, Dr. Tiffany.
All right. Thank you. Thank you for having me, and thank you for joining. I know we're a bit behind schedule, so I promise I'll try to stay on track, but I really want to get through everything because we have a lot to go through with the topic of optimizing implantation from the endometrial point of view. Just a quick disclosure, as you know, I'm the endometrial product director for Igenomix, part of the VitroLife group. So let's jump into it. So I tend to start my presentations with this bit of a striking statistic, being that we are well in 40 years of IVF in 2023. Our live birth rates following IVF are still only 25 to 30% per started IVF cycle worldwide. And we know that implantation is a significant rate-limiting step in both human reproduction and success in IVF. So implantation process hinges largely on two essential players, and I think you can guess who they are. One is the embryo, and the other is the endometrium. So for years, we have focused on optimizing the embryo in IVF. So we switched from standard insemination with IVF to ICSI, the way we culture our embryos in the lab, our incubation systems, how we cryopreserve our embryos and gametes, and lastly, the implementation of PGTA. Now, with that said, implantation failure still occurs in about 1/3 of PGTA cases. So even if we improve the embryo, there's still the other side of the equation that we need to look at. So that's what my talk is on today. So we know the endometrium plays a role in implantation from the microbiome to endometrial receptivity, all the way through decidualization. Today, though, we're going to focus on endometrial microbiome and receptivity portions of it. So starting with the endometrial microbiome, our knowledge of the microbiome in general is in large part to the Human Microbiome Project that was initiated about 16 years ago, with the goal to understand the microbial components of human genetic and metabolic landscape and how they contribute to normal physiology and the predisposition of disease. Some fun facts: humans have 10 times more bacteria than cells living in our body, especially on the skin and digestive tract. In a person who weighs 70 kilograms, about 1 kilogram is just purely bacteria. And between 20 to 60% of these bacteria, depending on the location, are not culturable under standard laboratory conditions. So since we're talking about IVS, let's hone in on the urogenital tract. This is about 9% of the total human microbiome, where we could see the dominant bacteria is lactobacillus. So we know from recent studies that the urogenital tract in females, the microbiota is a continuum throughout the reproductive tract, where we have the most abundance of bacteria in the vagina. However, it's mostly composed of lactobacillus, so there's very little diversity when we look at the vagina. Now, going up through the cervix to the endometrium, we get lower abundance of total bacteria, but the diversity increases with still lactobacillus as the dominant bacteria found. And that goes all the way up through the ovaries, where we see more diversity, but again, less biomass total. So we now know that the endometrium is not a sterile cavity as we had once assumed, and that the microbiome itself in the endometrium is quite different than what we see in the vagina. We know that Lactobacillus is the most abundant genus in the endometrium, so similar to the vagina. And we started exploring the effects of reproductive outcomes of the endometrial microbiome. So our group published a paper back in 2016 where patients were undergoing a personalized embryo transfer guided by ERA. But then we looked at the microbiomes of those patients, and we distinguished into two groups: Lactobacillus dominated, which was 90% more Lactobacillus in their particular bacterial profiles, or non-Lactobacillus dominated. So less than 90% of their microbiome consisted of Lactobacillus, and there was presence of other bacteria there. And what we found was that those patients with a Lactobacillus-dominated microbiome had significantly higher pregnancy rate per transfer, implantation rate per transfer, ongoing pregnancy rate per transfer, and live birth rate per transfer versus the non-Lactobacillus-dominated group. We also observed lower miscarriage rates, albeit did not reach statistical significance, where we had a rate of about 16.7% in the Lactobacillus-dominated versus 60, that's 60, in the non-Lactobacillus-dominated. So not only did Lactobacillus appear to be important in establishing the pregnancy, but also maintaining that pregnancy. So again, we look at the bacterial profiles from patients with live birth versus miscarriage or not pregnant, and we see in the live birth that it's highly distinguished by high percentage or presence of Lactobacillus compared to the miscarriage or non-pregnant groups. Looking at the negative outcomes as far as miscarriage and non-pregnant, we see other bacterial present, essentially pathogens, so Gardnerella, Streptococcus, and Staphylococcus were among the common ones found in the poor reproductive outcome group. So then we got a glimpse of the endometrial microbiome in early pregnancy. So this was unintended. So this was a patient with a history, you could see, of previous miscarriage and without successful live birth. Now, she was sampled at exactly about 4 weeks pregnant. So again, inadvertently, we were looking at the endometrial microbiome through an endometrial fluid sample. But this gave us the ability to look at what an endometrial microbiome looks like in early pregnancy. So we could see here in the miscarriage, she had very little presence of Lactobacillus and a high presence of bacterial pathogens, whereas in the pregnancy, we see a high percentage of Lactobacillus and very little other bacteria. Again, further supporting the claim that Lactobacillus is not only important in establishing pregnancy, but maintaining pregnancy. In 2022, our group published this paper right here entitled Endometrial Microbiota Composition Associated Reproductive Outcome in Infertile Patients. This was a prospective observational multi-center study with a sample size of 452 patients, and these patients were undergoing IVF or egg donor with personalized embryo transfer by ERA with a blastocyst of either day 5 or day 6 in an HRT cycle. As I mentioned, this was a multi-center study with over a dozen clinics participating worldwide. And all patients underwent a transfer at optimal time of receptivity as defined by the endometrial receptivity analysis test. So looking at the pregnancy outcome, we had a pregnancy rate of 58%, and out of that, 41% went on to live birth, 8% biochemical pregnancy, 8% miscarriage, and less than 1% ectopic pregnancy. So then we took the clinical outcome and we looked at the microbial status of those patients at time of biopsy. And we noted that those patients with a live birth had optimal microbes in their endometrial profiles versus those patients with poor outcomes as far as biochemical pregnancy, miscarriage, or no pregnancy. So the summary of our results here showed that pathogenic profiles is associated with reproductive failure, and many times this included well-known pathogens such as Gardnerella, Staphylococcus, and Streptococcus. Patients with a higher abundance of lactobacilli were more likely to achieve reproductive success. The endometrial microbiota should be considered as a possible emerging cause to implantation failure and/or pregnancy loss, especially with our knowledge relating to chronic endometritis, where we see that these specific patient groups at our increased prevalence rate of having these bacterial pathogens present. And the endometrial microbiota composition before embryo transfer could be a useful biomarker to consider, offering an opportunity to further improve diagnosis and treatment strategies. So looking at other clinic groups that have looked at the microbiome, this paper here is from Dr. Iwami of Japan, published in 2020, which was a prospective cohort study performed at her clinic and consisted of 158 women with recurrent implantation failure. The study group consisted of 107 patients who underwent the EMA test before an additional transfer, and 51 patients with a history of RAF that continued to transfer without any testing or intervention. Here, the cumulative clinical pregnancy rate in the study group after performing the test and any necessary therapeutic recommendations was found to be significantly higher than the control group, so 64.5% versus 33.3%. The implantation rate and ongoing pregnancy rate were also statistically higher in the study group versus the control group. So in the following years, this group went on to add to the data of the field and recently published the study, looking at endometrial microbiome and clinical outcomes in recurrent implantation failure patients. So in another prospective cohort study, this one consisted of a total of 195 women with RAF, and the primary outcome measured here was cumulative clinical pregnancy rate after 2 additional transfers. The results here showed that 1 in 4 patients needed antibiotics to treat a pathogen detected on the EMA test, and 90% of the antibiotics administered were not broad spectrum and were targeted to the specific pathogens detected. So this is one of the advantages of using a molecular technique to look at the microbiome versus, say, culturable or hysteroscopy or histology, is that we're able to actually identify which bacteria is present and then design a treatment based upon the patient's own unique bacterial profile. So that way, we're decreasing the risk of any recurrent infection in that patient. As far as the clinical outcomes here, statistically higher HCG positive rate, clinical pregnancy rate, ongoing pregnancy rate, and delivery rate was higher in the group that underwent the molecular testing through EMA with subsequent treatment if necessary versus the control group who proceeded with transfer without any testing. So just to give an update as far as the EMA test, as of this week, there's been a change in technology to RT-PCR, referred to our upgraded EMA and ALICE. The Igenomix microbiome test EMA and ALICE were launched in 2018 using next-generation sequencing for a complete view of the endometrial microbiome, which was poorly understood in that moment. So keep in mind that the previous studies we just reviewed were utilizing this technology. Since the launch, we have analyzed more than 70,000 clinical samples. And as a reflection of our experience with the test with NGS, we now know which bacterial pathogens are most frequently detected in the human endometrium. So this was something not known just a few years ago. Based on this data, we've designed an exceedingly precise and complete panel that contains 97.8% of the bacterial pathogens detected with NGS, and all bacteria included are clinically relevant, and most are identified at the species level, allowing for more targeted antibiotic treatment. So finishing up with the conclusions pertaining to the microbiome, the vaginal and endometrial microbiomes are different. Lactobacillus is the most abundant genus in the endometrium. A lactobacillus-dominated endometrium is associated with a better reproductive prognosis in IVF patients. The presence of pathogens in the endometrium is associated with implantation failure, recurrent miscarriage, and preterm birth. And finally, the endometrial microbiota composition before embryo transfer could be a useful biomarker to consider offering the opportunity to improve diagnosis and treatment strategies. So we talked about the endometrial microbiome, which is kind of the first barrier that the embryo encounters at implantation. And hopefully it's optimal in order to improve implantation. But beyond that, we need to consider endometrial receptivity. So let's start with the menstrual cycle. Obviously, we know there's various changes throughout, with varying biological functions activated accordingly. But the embryo can only implant in the endometrium with a receptive phenotype. And in most women, this coincides between day 19 and 21, which is referred to as the window of implantation. The window of implantation was first proposed in the mid-1950s, with various lengths defined since then. So earlier studies suggesting up to 10 days, later studies said about 2 days, and now molecular studies are showing a bit of a smaller window between 29 to 36 hours. But ultimately, the window of implantation concludes upon initiation of stromal cell deciddualization, subsequently permitting trophoblast invasion and placental formation. So when is the best time to transfer an embryo in an IVF cycle? We should be asking not if, but when. So typically, we expect endometrial receptivity, as I mentioned, between days 19 to 21 in a normal menstrual cycle. This correlates with about 5 days of progesterone exposure, whether it's in a natural cycle or an HRT cycle. So when we're performing IVF, HRT, we'll perform, again, after about 120 hours of progesterone exposure, or a week post-LH surge, or 168 hours from HCG trigger. How do we prep the endometrium for IVF transfer? Well, a lot of our preparation assumes that the window of implantation is constant in all women. However, in fresh IVF cycles, we need to consider the potential effects of supraphysiological steroid levels in controlled ovarian stimulation and the deterioration of endometrial receptivity. But in most cases, we monitor with hormone levels, specifically estradiol, progesterone, maybe LH if we're doing a natural cycle. And we rely on the assessment of endometrial thickness by transvaginal ultrasound. Now, going beyond that, historically, there have been other methods of either inducing or finding receptivity. One is endometrial scratching. However, this is not widely performed, I would say, this day and age, especially following a Cochrane review showing that it's not effective. Histological dating was previously used, but again, there's some inconsistency and the subjective matter. This method is not typically used. And then lastly, biochemical markers have been proposed, but however, this usually consists of maybe 1 or 2 biochemical markers. And in the case of endometrial receptivity, being that it's such a complex process, it's impossible to define with a single or a few biochemical markers. So that comes to the introduction of transcriptomics, which is a study of the full range of messenger RNA molecules expressed from genes of an organism. The analysis of gene expression patterns in particular tissues under distinct conditions allows not only for the diagnosis of pathological conditions such as cancer, but also physiological processes such as endometrial receptivity. And a prime example of this is the endometrial receptivity analysis, or ERA test, which was the first commercially available transcriptomic test looking for the window of implantation. This test analyzes by next-generation sequencing the expression of 248 genes related to endometrial receptivity to determine the optimal day for embryo transfer for each specific patient. An actionable directive for a more personalized approach to embryo transfer is provided to enhance synchrony between the implanting blastocyst and a receptive endometrium. In fact, we see here the gene expression pattern associated with the window of implantation, where we see an onset of genes turning on, in which we refer to as a transcriptional awakening. So as far as the personalized embryo transfer guided by ERA, this is going to depend on each patient's own time of optimal receptivity. So in the first example, this patient actually needs less progesterone exposure since she reaches the window of implantation earlier than expected. We have patients transferring at the expected time of endometrial receptivity, so P+5, or approximately 120 hours of progesterone exposure, which is still a personalized embryo transfer. This is a wild misconception that patients who are found to be receptive at the standard time are not undergoing a personalized embryo transfer, but indeed they are, because we have to consider the preciseness of the recommendation. And then a patient who is quite pre-receptive, needing an additional 48 hours of progesterone exposure. Now, the ERA test was indicated and still indicated for patients with recurrent implantation failure. In one of the first studies, we looked at those patients with RAF versus a control group, so 0 to 1 previous failure. And what we saw was that approximately 1 out of 4 patients with RAF had a displaced window of implantation at the standard time of embryo transfer. However, when adjusting for this displacement and transferring a time of indicated optimal receptivity, clinical outcomes were similar between the RAF and the control groups without any statistical difference. In a subsequent study, a case report of a patient with 7 previous failed cycles. You could see here she utilized fresh, frozen day 3, day 5, and even utilized an oocyte donor in her last 3, never became pregnant. She underwent the ERA test and was found to be pre-receptive by 2 days, needing an additional 48 hours of progesterone exposure. Undergoing a personalized embryo transfer on P+7, the patient transferred 2 blastocyst embryos and subsequently gave birth to healthy twin boys. And then, much more recently, in an external paper by Jia et al. In 2022, they compared the effectiveness of ERA-guided personalized embryo transfer with traditional FET in patients with RAF. In this study, they enrolled 281 patients, and the primary outcome was pregnancy and implantation rate after 1 embryo transfer. Here we can see the patients that underwent a personalized embryo transfer guided by ERA had statistically higher implantation rate, pregnancy rate, and clinical pregnancy rate versus the FET group who underwent transfer at the standard time. The conclusions here was that offering ERA to patients with RAF can be effective in improving implantation and pregnancy rates compared to traditional frozen embryo transfer. In another recent paper, a meta-analysis by Liu et al. From 2022, they had the objective to assess the prevalence of a displaced window of implantation in infertile women and the clinical utility of personalized embryo transfer guided by ERA in IVF-ICSI outcomes. Their conclusions were this. Considering the approximately 1/3 of infertile women could suffer from a displaced window of implantation, the ERA test emerged as a promising tool. So although their meta-analysis demonstrated that patients with general good prognosis may not benefit from ERA, a PET guided by ERA significantly increases the chance of pregnancy for non-receptive patients with RAF of endometrial origin. In fact, we can see here when we compare the clinical outcomes for good prognosis patients versus RAF, we saw similar outcomes. So again, normalizing outcomes for patients who've had repeated implantation failure. So I focus on RAF as that's the indication for ERA. And I think everyone's probably familiar with the paper published from Shady Grove at the end of 2022. They conducted a randomized controlled trial in good prognosis patients, so again, patients without recurrent implantation failure. And in fact, they further validated the results that our group did in RCT looking at the same good prognosis group, in which we didn't see statistical difference between patients who underwent ERA versus those who underwent the standard transfer time. However, in the RCT from Carlos Simon 2020, they went a step further and looked at the cumulative live birth rate, in which a statistically significant improvement in live birth was seen in the PET with ERA arm versus both the standard FET and standard fresh embryo transfer arms. So we did speak with Nicole Doyle from Shady Grove, and they are continuing the study looking cumulative live birth rate, so we look forward to that. Now, as I had mentioned, one thing we need to focus on is the indication for ERA, so RAF. But another thing is the protocol compliance within these studies. So even in our own study, we were not immune to poor protocol compliance. In fact, we had a 50% dropout rate, either because patients didn't replicate the cycle or the recommendation wasn't followed, so forth. Now, in the Shady Grove paper, they had a very low dropout rate, but regarding protocol compliance, one thing that we did know was a deviation in proper endogenous progesterone control. So it's imperative that the protocol is followed exactly in order to ensure best results for your patients. Looking back at our own study with our 50% dropout rate, we actually extracted data from one clinic that participated in the study that had published a paper on their retrospective review late last year, in which we found this group had a 36% dropout rate due to poor protocol compliance, and this was within a clinical trial. So again, it's important to highlight that protocol compliance is necessary, not only in order to ensure good study results, but also good results for your patients. So the conclusions here: 1, the endometrial factor plays a key role during implantation process, and we cannot always solely rely on the embryo for success. 2, although most of women are receptive at the standard window of implantation, some of them reach the receptive phenotype earlier or later, showing displaced window of implantation. 3, to increase the probabilities of getting pregnant, it is important to synchronize a competent blastocyst with a receptive endometrium, through the personalization of embryo transfer. And then my last conclusion here is reader beware. Proper analysis and protocol compliance should be considered when reviewing publications pertaining to endometrial receptivity, whether that's with ERA or any other test. So with that, I know I went over time and I apologize, but I want to thank you for your attention and for inviting me to be here today.